Good morning and welcome to the Nusano KOL event. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal presentations. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call is being recorded and a replay will be made available on the Nusano website following the conclusion of the event. I'd now like to turn the call over to Chris Lowe, Chief Executive Officer at Nusano. Please go ahead, Chris.
Good morning, everybody, and welcome. Thank you for joining us today. My name is Chris Lowe. I'm the CEO. We have a great agenda for you today that I'll walk you through. First, we're going to have a couple of really exciting speakers, a couple of KOLs, Dr. Pomper and Dr. Atcher, who have agreed to join us, and will be talking about the next opportunities as with regard to radioisotopes and the evolution of this space and how this enables really what we think is the next generation of radiotherapeutics. After that, we'll have a great Q&A session and wrap up within the hour. In addition, during the Q&A session, both myself, Dr. Pomper, Dr. Atcher, and our co-founder and COO, Dr. Howard Lewin, will also join as well. Next slide. To introduce Dr. Pomper, we're very fortunate. Comes to us from Johns Hopkins, where he's the director of nuclear medicine and molecular imaging. Really just a world-renowned expert and a lot of experience in this space, both in terms of the development of new radiopharmaceuticals, as well as other techniques or breaking techniques for molecular imaging of cancer and CNS disease as well. Next slide. And then in addition, we also have Dr. Atcher from the University of New Mexico, again, a world-renowned expert in this space, the author of over 100 publications, numerous patents and scientific abstracts as well. His work has really touched on some of the thought leaders in the space and some of the best in terms of hospitals and institutes where he's performed his research and practiced for over 40 years. Next slide. So just very briefly about our company, about Nusano. As we know, the current production methods for radioisotopes just frankly are insufficient for supporting the demand that we know is coming in this space. We're leveraging 1940s to 1970s technology, and the supply chain is extremely stressed in that a lot of the supply material originates in Russia. It's a very Russian-dominated supply material, source material market. And what we do at Nusano is we're able to break through and get us off of all of those problems. So number one, we have a source technology which generates heavy ions, resulting in just a game-changing increase in yield and efficiency of production. Our core technology, the beam that creates this production, it also enables just a broad array of isotopes. So when we come to market in Q1 of 25, we believe we'll be able to produce up to 40 different radioisotopes, and we think that that'll be a key enabler of the next generation of radiotherapeutics. Finally, with regard to our core technology, we now have over 10,000 hours of experience with it. We know that it really only requires a few days of preventative downtime per year and has the ability to switch from one isotope to another in a matter of 10 to 15 minutes. So we believe that we're bringing a game-changing technology to market, which will be able to enable a lot of really interesting opportunities in the development path of radiotherapeutics. Next slide. And so lastly, this plant is not a dream. It's well on schedule. As I mentioned, it's open. It's located just south of the Salt Lake City airport. This is facility 1 of 4 that we expect in the US. It's about 170,000 square feet in total, about 100,000 square foot footprint. And you can see some recent pictures of it. We feel very, very pleased that this facility 1 will be enclosed and weatherproofed by the end of this year. We'll have heavy engineering work next year and be ready to start the production ramp in Q1 of 25. So with that, let me go ahead and let's get on with our core presentation here and go to the next slide and allow me to introduce Dr. Pomper. Dr. Pomper, please take it away.
Great. Well, thank you very much. And thanks to the organizers for inviting me to speak today a little bit about radiotheranostics for cancer. It's a new way to manage a variety of cancers. Some disclosures. So by way of outline, first I'll give a brief introduction. I'll then talk about some of the current agents, some emerging agents, and then provide a perspective at the end. So when we think about precision medicine, we often think of that as a genetic phenomenon where the patient's genes or their epigenome might dictate what kind of new therapies that they're going to get. These are very specific therapies for very specific targets. But I sort of look at the genetics as on-the-ground intelligence about the patient, while the imaging actually provides you the ability to develop guided missiles to hit very specifically targets within the body. It gives you spatial information, and that's one half of the theranostic paradigm. So an example of a precision imaging agent might be this positron-emitting compound called DCFPYL, which is now known as Pylarify. It's used to detect prostate cancer that expresses the prostate-specific membrane antigen. So it's a very specific target. So here we have actually two patients, patient A and B, and these patients have basically the same disease. So they go to the doctor because they had a rising prostate-specific antigen level in their blood, the blood test for prostate cancer, and the physician performed a rectal exam and felt a lump on their prostate. They had a biopsy, and they were both determined to have high-risk prostate cancer. However, although they seem the same in that regard, when we do the imaging, we can see that they're actually very different. So the patient A has localized disease, will likely be managed by surgery, whereas patient B has disease that's already left the prostate, has metastasized, and will get some sort of more systemic therapy, such as a endocrine therapy or a chemotherapy, or maybe a theranostic agent, which is also generally used for more systemically distributed disease. So let's talk for a second about theranostics. So theranostics is not a new concept by any means. In fact, what it really means is just having a diagnostic biomarker to lead what is going to be done therapeutically in the patient. However, for our purposes, we will stick with the radiotherapeutics or the radiotheranostics in the context of what we're going to be discussing. So if you look on the right, you could see that the theranostic basically has three different components. It's got a radionuclide, which can be either for imaging, it could be a positron emitter like fluorine-18, as I showed earlier, for positron emission tomography, or it could be for therapy. So that's sort of where the idea of theranostic, therapeutic, and diagnostic comes into play. And for therapy, we could have beta particle emitters or alpha particle emitters, and there are other particles that can also be used. Then there is a linker, which is part of the theranostic that links the warhead up to the affinity agent or the binding molecule that binds specifically to the target cell, in our case, to the cancer. So three parts to a typical theranostic agent. So here we see a number of the more famous radionuclides that are used. For this purpose, there are beta particle emitters shown here, as well as alpha particle emitters. The main issue here is that the beta particles tend to travel further in tissue upon decay of the radionuclide than the alpha particles do. And that becomes important for reasons that I'll discuss in a few minutes. Now, theranostics, as I mentioned, it's not an entirely new concept. It's not totally dedicated to the radiotherapeutics, but it has been around for at least 80 years. So back in 1934, Enrico Fermi actually discovered the production, or he could induce radioactivity and produce isotopes such as iodine-131, which is a very famous and the first therapeutic radionuclide. Not many years after Fermi did, and by the way, he got the 1936 Nobel Prize for that discovery. About 10 years later at Harvard Medical School, Saul Hertz and Arthur Roberts started using I-131 to treat Graves' disease. It was then used to treat cancer after that. And we still use the exact same agent in the clinic today, many years later. If we come into sort of the so-called modern era, which is now already 40 years ago when I started graduate school, we were, this is the very first paper that I read as a graduate student. We were developing steroids, estrogens that were labeled with I-125, potentially to treat estrogen receptor positive breast cancer. So modern theranostics are really kind of along this line. Even though this is 40 years ago, what makes things different now is the fact that we have a lot more isotopes to use. We have more affinity agents. The chemistry is easier. The detecting systems, the PET scanners are much better. And as you heard in the beginning introduction, there are new ways to produce isotopes and have them distributed so that we can actually use some of these things for medical purposes. So in terms of current agents, as I mentioned, there are the thyroid agents. There are other agents that we're using every day for neuroendocrine tumors, but I'm going to focus more on the prostate cancer agents that target the prostate-specific membrane antigen. That's sort of my area of expertise. This article was written by one of my colleagues, George Skouros, 3 years ago already. And you can see that even then, there are quite a few radiotheranostic agents that were either in use or under clinical development. I would hazard a guess that this has probably doubled in the last 3 years. So prostate cancer is a major public health problem. 1.3 million cases, 416,000 deaths globally. It's got the highest incidence of all cancers in men in 114 countries, leading cause of cancer death for men in many countries. And if you have metastatic bone disease, you've got a 40% one-year survival rate. So very serious, very lethal cancer as well. So this sort of shows the events that occur during the journey of a patient with prostate cancer. The stars, A through D, represent where imaging might come into play. So at diagnosis, one wants to know just where the disease is so that we can, dictate the therapy. So you'll image at that point, and then there's surgery, et cetera. And then eventually, and the reason that this is already so far into the journey is the use of these theranostic agents. However, that is hopefully going to be moved up earlier in this clinical course because they're beginning to show a certain amount of efficacy. So at first I thought that alpha particle emitting agents should be used for micrometastatic disease because they don't really travel very far, whereas gross recurrent disease, big tumors, as shown here on the left, would better be served with a beta particle emitter, which travels further. You can get around things a little bit better, but that doesn't really matter with the affinity agent that you're using. And you can use either an alpha or a beta particle emitter, depending on whether or not the patient has for example, shown any kind of response to the beta emitter, which is usually used first. The more important issue is that the alpha particles have what's called high linear energy transfer. So they deposit their energy along a very, very short path length, as I showed earlier. These are helium nuclei. They're like hand grenades, very lethal. One decay event can kill a cell, very hard for cells to work around alpha particle emission therapy in terms of mutating to get away from it, but it still does. This just shows a typical theranostic agent. You've got your warhead held by a chelator. You've got a linker, which could be used to modify the pharmacokinetics of your compound once you inject it. And then you've got your affinity agent that binds specifically to the cancer target. So here we see 6 patients who have prostate cancer with varying amounts of cancer shown in red from a PET scan. These numbers are the PSA levels. So some patients with a lot of cancer have PSA levels of 1,000. And you could see that by using beta particle therapy, and this is already 5 years ago, that one could eradicate almost all the visible cancer, all the PSMA positive cancer in the patient. So the patients can then, this patient, by the way, was wheelchair bound and now can resume his normal activities. So another huge benefit of these therapies is the vast improvement in the quality of life of the patients. The first prospective trial, and I focus on prospective because those are the real clinical trials that you can really start thinking about the utility of a new agent. The first prospective trial was done in Australia, and you could see on this waterfall plot, the blue shows the decrease in PSA after 12 weeks of therapy. And the blue is actually 50% decrease, and anything below 0 is a decrease. So if you go to the best response, you see everybody had a pretty good response. In fact, there was objective response in 82% of the patients that received this therapy, which was quite heartening for the therapy. The same group did another trial where they compared the theranostic agent to standard cancer chemotherapy, cabazitaxel. Same sort of thing. We have a waterfall plot, except this time in red, we see the reduction of greater than 50%, or we see a reduction greater than 50%. In fact, a little bit more so in the theranostic case. And if we look at the sort of the timeline over 18 months, there was a sort of an enhanced progression-free survival out to 18 months in this trial, which was published a couple of years ago. But still, the Kaplan-Meier curves are dishearteningly close. So we're still not doing, we're only maybe months better, if that, using the theranostic agent than the chemotherapeutic agent, but with far, far fewer side effects and very few adverse effects during the trial. Now, to bring an agent like that to the clinic, we have to do what's called a registration trial, which is a phase 3 trial where you've got hundreds of patients at multiple different centers. So this trial called Vision put patients who were on the standard of care therapy plus theranostic and compared them to just standard of care therapy alone. And what was shown is that there was not only an imaging-based progression-free survival, so you use imaging to look at the progression, and there was better progression-free survival in the patients that received the theranostic, but also the key, in fact, the reason this was published in the New England Journal of Medicine, the key outcome indicator of overall survival was better for the theranostic agent than standard of care. And what this shows is not just that the survival is better, but again, very few side effects. So I have many patients, I'm a radiologist, but I have many patients calling me to try to get this therapy because they don't want chemotherapy going forward. So in terms of emerging agents, and this will be covered largely in the next talk by Bob Atcher, but I'll just touch upon for a moment 2 of the more famous ones, the actinium-225 and the astatine-211. These are alpha particle emitters. I believe that the field will pivot to a certain extent toward these more energetic and effective agents. So there are key differences, however, between the two. So the actinium-225 has a much longer half-life than astatine-211. However, interestingly, I've read that some of the agents that have been made with actinium-225, because there are daughter nuclides, in other words, I should say daughter alpha particles, shown back on the previous slide, if I can get back to that. I guess I can't, but in any event, there it is. So you can see that there are other alpha particles that are given off. one does not have control over those alpha particles, because once the first decay occurs, the molecule falls apart and these other alpha decays occur elsewhere in the body. Astatine doesn't have that issue. There's only one alpha particle that's emitted. So even though there's a 10-day half-life, these actinium compounds are not necessarily very stable in solution. And, there may be issues with shipping, which is why it's important to have these sites around the country that can be used so that we can use the materials quickly after they're synthesized. The other thing I want to mention is that all the agents other than astatine-211 are produced from radium-226. So we have to consider ease of radiochemical, or I should say radionuclide incorporation into the molecular species that you're using to hit your target. We have to think about the physical half-life, the synthetic yield. This absence of daughters is preferred, stability for transport, availability of the nuclide, and then, of course, the cost. So those are things that we have to consider. This is an example of using actinium after a beta particle emitter fails. You can come in with an alpha particle emitter and you can eradicate tumor. The only trouble is that these alpha particle emitters are quite powerful, and you can also eliminate normal tissues. These are salivary glands. These are not malignant tissues. So the patient will have to use a squirt bottle basically the rest of his life. This is kind of a showstopper in the United States. We can't do trials like that, and we need workarounds for those kind of off-target issues. So we can either synthesize compounds that don't hit the salivary glands, or we can do other things. We could use gentler agents. Nevertheless, there are 22 studies that are currently ongoing that are using actinium-225 based theranostic agents. But we prefer sort of a kinder, gentler, a tamer alpha particle emitter with only one alpha coming directly from the astatine. So one particle, there's no daughters, and we can actually see the astatine-211 by virtue of X-rays that are produced. But there are some challenges. The availability currently is somewhat limited, hopefully to be offset by entities such as Nusano. It's got a relatively short physical half-life. There's no stable isotope for preclinical development. It will be easier to have that. The stability can be a little bit decreased sometimes because of the astatine carbon bond, and the compound can deteriorate a little bit, but there are ways around those things that we're working on. It's inexpensive to produce. However, you can produce it from natural bismuth. Here's a Pepto-Bismol tablet can be used for producing astatine-211. The targeting, et cetera, is already existing. However, one does need a high energy cyclotron. There are lots of cyclotrons in the US, but not a lot of cyclotrons that are 28 mega electron volt. So those need to be expanded, and there are other ways of doing that besides the cyclotron. This just shows that we can actually see the astatine if we need to use that for dosimetry or other purposes. So there are existing accelerators in the United States. In Japan and in Europe, there's a network to make astatine specifically more available because of its perceived utility. There are new accelerators that are coming online in both commercially available and ultra high beam current accelerators, such as the one that's sponsored by Nusano. So our group, and Michael Zalitsky at Duke University, have made a number of astatine compounds to date. These are our so-called second generation compounds, but I just want to mention that some of the work that we've done, is shown here circled in red, has been recognized as the most significant basic science paper in its respective journal for that particular year, highlighting not just how important astatine is, but how important the theranostics is going to be going forward for the field of nuclear medicine. So with that, let me just finish by saying that we have to think about number one, the availability of the isotopes, which hopefully will be offset fairly shortly. We need to think about who should get this therapy, what the dosing schedule should look like, if there are intelligent ways of developing combination therapeutics. We are beginning to use artificial intelligence to predict what patients, based on their images, might respond. We could look at their blood values and see which patients may be too frail for this therapy. We have to worry about the heterogeneity of the tumors, because they will eventually somehow work around even these vicious alpha particles. And then we have to worry about long-term toxicity, which has implications for which compound you may want to use and how many times you can give it, et cetera. And with that, I just want to thank my team. I consider this not my research group per se, but a coalition of experts that we have at Johns Hopkins that are working on these things, as well as our PET Center, and then, of course, the patients and their families. So thank you very much for that, and I guess I'll hand it off to Dr. Atcher.
Thanks, Dr. Pomper. Let me do a little bit more of a deep dive in terms of the availability of the isotopes, because obviously without an isotope, none of these targeting agents have any or much impact at all. So on the next slide, I'll remind you of the list that Dr. Pomper put up to narrow down the isotopes that we might be interested, or that are currently of interest in terms of developing these therapeutic agents. With the beta emitters, yttrium-90, which has been used extensively for, I'd say, about the last 20 years, has limitations, though, because if it gets free, it goes to the bone, which means that the bone marrow becomes very important in terms of normal tissue toxicity. As Dr. Pomper mentioned, iodine-131 has been around forever. Its great advantage is that the thyroid tissue has a great affinity for the iodine, so you're able to localize a very high percentage of the injected dose and achieve therapy for thyroid cancer and Graves' disease. Samarium-153 was developed to address bone metastases. It's a beta emitter that has had some success in its application. And then we'll talk a lot more about lutetium-177, which has been of interest and extensive interest recently as a potential therapeutic. Dr. Pomper did a really nice job of outlining the alpha emitters that are of interest and under development. When I was putting this slide together, I realized that for radium-223, my thesis, which I started in 1976, had work on radium-223 as a potential therapeutic. So gives you an idea of how long some of these isotopes have been around before we've actually been able to get them into the clinical trials and hopefully the clinic. Bayer Chemical has been using radium-223 as a therapeutic. Its name is Xofigo. And it's one of the things that I love about this field. We make unpronounceable materials so that you have to be a part of the secret society in order to be able to talk about any of these things. So I'll get into more detail on the ones that that Dr. Pomper mentioned, as well as under more extensive development. Next slide. So this will just give you a picture of the three or the two different particles that we've been talking about. There is a track for a low energy electron and a high energy electron. And this is the track is demonstrating an ionization tract. The ions that are produced are what do the damage to the cell to kill the cell. And so when you're looking at these images, you can see that there's spaces between where you see an ionization event, and that has implications in terms of the ability of an electron to kill the cell. In contrast, down in the bottom left-hand corner, you see this intense ionization tract that's created by the alpha particle. And as a result, any traversal of a cell that you have with an alpha particle, you're almost inevitably going to get a cytotoxic event. Next slide. So in terms of the biologic effect with the beta particles, as Dr. Pomper mentioned, there's low linear energy transfer. You saw in the prior slide, excuse me, that those ionization tracts have spaces between the events. And as a result, the toxicity of a beta particle is dependent on how much dose you can deliver quickly. You want to get a dose rate that's very high. The reason for that is that the cell has a capability to repair the damage that's done. It's something that keeps us alive, because anything that is toxic to the cell and creates DNA breaks are potentially repaired. So you really have to have a pileup of those events with the beta particle in order for it to be successful. In order to have those ions produced, you also have to have oxygen present. And so those parts of the tumor that have low oxygen or no oxygen are much more resistant to treatment with the low LET radiation, whether it's a beta particle or external radiotherapy. And as I said, it takes multiple transits to kill the cell, up to 20 if you look at the dosimetry calculations that are done by my colleagues in medical physics. And the other thing is you could see in that prior slide, the range can be up to 1 centimeter. So that you have a long distance from where the radioactive decay occurs, that you have travel of the beta particle. In contrast, the alpha particles have the high linear energy transfer. Because they are so densely ionizing, they create damage to the cell that's not repairable. So their toxicity is dose rate independent. So you don't have to have that pileup, that quick pileup of dose in order to create a cytotoxic event. In addition, the presence of oxygen is not necessary. And we've done studies and published studies that indicate that cells that are lacking in oxygen are still able to be killed by the alpha particles when you compare it to the low LET radiation, where the cells can grow through it. You only need 2 to 3 transits in order to kill the cell. So when you're delivering these labeled materials, you have to have 20 or 30 radioactive atoms in order to get a cytotoxic event, whereas you only need 2 to 3 radioactive atoms in order for the high LET radiation to be successful. And the contrast to the beta particles, the alpha particles have very intense tracks of ionization, and the range is only up to about 100 microns. Next slide. So how do we produce these radioactive materials? Nuclear reactors in the US, the University of Missouri reactor has been very active in terms of production of radioisotopes, and is responsible for many of the ones that I've discussed here. The national labs have 2 reactors, Oak Ridge National Lab and Idaho National Engineering Lab. These are very much higher energy than the Missouri reactor, but their operation is not continuous as it is with the University of Missouri. And so you've got issues associated with their operating schedule relative to output of the isotopes of interest. There are other academic research reactors that are useful for isotope production. And the company Shine, located in Wisconsin, has been using a different technique to produce the neutrons, and they have been active in producing lutetium-177. It's not limited to the US. There are facilities in the Netherlands, Belgium, Canada, South Africa, and Australia, which have been optimized for isotope production and are active and have been for some time in terms of production of medically useful isotopes. In contrast with the accelerators, there are high energy accelerators at the national labs, but we have the same problem in terms of operating schedules in that they don't operate 24/7, 365. They have operating schedules which are roughly half a year, and there's a lot of effort to try to make sure that the accelerator at Los Alamos is running when Brookhaven isn't, and vice versa, so that there's some continuity in terms of their ability to supply useful isotopes. There are companies which have been instituted in the last a few years that are purchasing commercially available cyclotrons and setting up to produce isotopes that are of interest to both the imaging and the therapy applications. As Dr. Pomper pointed out, there are academic cyclotrons that are also involved in radioisotope production and linear accelerators, and there are two companies, Isonetics and Nusano, which are in the process of developing new and better linear accelerators to address isotope production needs. In addition, there's international facilities that are involved in medical isotope production in Canada, Russia, and South Africa. So it's something that we're using worldwide capabilities to produce useful isotopes. Next slide. So there are advantages. The research reactors, very powerful, can produce huge quantities of material. The size of the targets are not, well, let me back up. If you want to double the size and production of a given radioisotope, there's a minimal increase in the cost of the isotope because you're not changing anything associated with the operation of the reactor. You're just using a little bit more space in that reactor. The bigger reactors happen to be multi-mission and have activities that are outside of isotope production. And so the operating funds for those facilities come from multiple sources. And rarely is the isotope production the dominant source of funding. So when some of the other programs decide that they're not interested in continuing their operation, it puts the isotope production in danger of those facilities. The accelerators vary in their capabilities. The cost of acquisition of an accelerator for those companies that are being set up to produce radioisotopes are manageable when you compare it to what it would cost to build a reactor to achieve some medical isotope production solely as its function. The cyclotron operation is totally controlled by any of the accelerators that we're talking about, with the exception of the high energy machines at the national labs. They're totally controlled by the user, so you don't have to worry about other programs that are involved in operating schedules, etc. And you get with the material that you produce has very high specific activity, which means that the percentage of the atoms that are in the product, a very high percentage of them are radioactive. Very few of them are stable and therefore don't produce any advantage in terms of their therapeutic or diagnostic capability. Next slide. There are limitations of both the production schemes. In contrast to the accelerators, the specific activity of the reactors is low. you put in a target of a given material, for example, the lutetium, and you have neutron capture in a small percentage of the material that you've put into the system. And so there's a very high percentage of your target material that's never activated. And so when you think about, I'm going to then make synthesize a material attaching these isotopes to a targeting molecule, you are not optimizing the number of target molecules that have a radioactive material associated with them. With natural abundance, many elements have a number of different isotopes. And so you get co-production of other isotopes, which in some cases limit the applicability of this for medical applications. If you use an enriched target in the specific target isotope that you're interested in, the cost of the target increases substantially. And as Dr. Pomper mentioned, there's limited production sites. Most of all, for many of these elements, the Russians are the sole source of the material. And if you have the co-production of other isotopes, sometimes it creates problems with long-lived waste, which generate regulatory hurdles in terms of cost, bonding, and disposal. You have, with the accelerators, the same issues if you use a natural abundance target. You co-produce other isotopes, which may or may not have an impact on its applicability for medical applications. You have the same issues if you use an enriched target, in that the target cost increases substantially and there's limited availability of material. If you are successful in creating high specific activity material, it generates minimal waste management issues in contrast to the reactor material. And the other problem is that if you want to double the amount of material that you're creating, you basically double the cost of the production. Whereas, as I mentioned, with the reactor material, you don't increase that cost in a similar way, because the reactor will operate whether you put in X amount or 2X amount of your target material. Next slide. So for lutetium-177, it's produced in a reactor. If you produce it by putting a lutetium target in, has a very high cross-section. So the number of lutetium-177 atoms that you produce is, relatively speaking, high. It has a convenient half-life for the logistics of distribution, synthesis, and final distribution of the radiopharmaceutical. And there's a separate and different way to produce it, where you produce a radioactive isotope of ytterbium, and then you separate the lutetium from the ytterbium, and you generate a much higher specific activity on the order of what you would from an accelerator. And the conjugation chemistry, is Dr. Pomper discussed, is mature, and so that's not an area that needs to be, you don't need to do much in terms of development. The disadvantages with the neutron capture is that the specific activity is low. You co-produce an isotope that creates some problems in terms of its disposal and other regulatory hurdles for its use. Next slide. So for the alpha emitter actinium-225, Dr. Pomper did a very nice job of describing some of the issues in terms of its clinical application. The best way to do it is to separate the actinium from the thorium-229 parent, which enables you to basically produce a pure actinium-225 product. You don't have any other isotopes of actinium, which creates some problems. You deliver, as he's pointed out, very high radiation dose. It's not so radioactive that you have to use very heavily shielded facilities. You can do most of your work in a glove box and do manual manipulation of the production and labeling activities. The half-life facilitates logistics in terms of distribution of the actinium, synthesis, and then distribution of the final product. There's other production options that are advantageous, and the conjugation chemistry, again, is mature. One of the disadvantages is that there's a limited supply of thorium-229, and in order for there's a lot of thought going into how we could possibly produce more of it, because it produces such a pure version of the actinium-225. There's been a proposal to use very high energy accelerators to produce the actinium-225. But the limitation there is that you also produce actinium-227, which has a 20-year half-life, and then creates some significant problems because chemically it will behave the same as the actinium-225. And so you're injecting a much longer life material that can contribute to a dose in the patients that is not necessarily to the target. And of course, in those instances, FDA approval is going to be much more difficult because you have to demonstrate the 227 does not have deleterious effect on the long-term effects on those patients. It also creates waste management issues because of its long half-life. A second way of making the actinium-227 is to use medium energy accelerators and use a radium-226 target. Just from the get-go, the radium-226 is in itself radioactive. So you're dealing with a radioactive target that you're going to use to make the actinium-225. The radium decays to radon, which is a gas, and so you have issues with containment of the gas that is generated from the target itself, as well as dealing with a target and material that creates some serious issues relative to potential contamination. And so this material needs to be worked up in a shielded facility rather than in a glove box, as you can do with the astatine. Again, you have the issue of redistribution of the daughters to non-target tissue, which Dr. Pomper mentioned in detail. And there's also paranoia around alpha emitters. I've used them in a number of different facilities, and it takes a little while to make everybody comfortable with the operation because there's such a concern about the introduction of alpha emitters into some of these facilities. Next slide. Dr. Pomper did a really nice job of describing the advantages of astatine-211, its accelerator production. Conjugation chemistry, again, is developed. The short half-life has an advantage in that the waste generated is is easily managed. And if you have any kind of contamination with a 7-hour half-life, after 3 days, your facility is again usable, so you don't have to shut down a lab or a building in some cases because of distribution of the radioactivity. The FDA has familiarity with the isotope, so if you are interested in developing a radiotherapeutic with that isotope, the major concern is going to be about the targeting molecule, not about the astatine or the linkage chemistry. And as Dr. Pomper mentioned, there's an expanding market to supply the accelerators that are used to produce them, which, as he pointed out, the current population is somewhat limited. Those cyclotrons that are at those academic centers, for example, are limited to only be able to produce about 6 doses per day. So very limited capability in terms of addressing a market that would have a large number of potential patients. The half-life presents distribution challenges. So you're really talking about a situation in which you want to have a regional center that produces material within a reasonable distance of the sites that are using it. The conjugation chemistry is pretty mature, but there's always the astatine is a very interesting element because it's got some characteristics like iodine and bromine above it on the periodic table, but it also has some others that are more metallic. And so there's been some thought that there may be a different system completely to be able to conjugate it to the targeting molecules. You will hear that there are cyclotrons around, about 200 cyclotrons around the country that are used to produce PET isotopes. But because they only accelerate protons and not helium-4 nuclei, they're incapable of producing astatine-211. Next slide. Dr. Pomper mentioned lead-212. It's similar to astatine in that it's a single alpha particle emission. The supply of the parent isotope, radium-225, is adequate to be able to meet the needs and is more easily expanded than what you saw with the thorium-229. The half-life supports doing chemical manipulation. You can actually produce a system in which you distribute the radium-224, and then on the site of doing your conjugation chemistry, you separate the lead-212 from the radium and then do your synthesis. So you don't have to worry as much about the distribution of the isotope before you begin your synthesis of the radiopharmaceutical. It's a short life, half-life, so you don't need to deal with some of the regulatory issues. And the supply of the thorium-228 to produce the radium-224 is abundant. The disadvantages, the 10-hour half-life limits distribution. The radium-224 has a 3.5-day half-life, so you basically have to resupply the site on about a weekly basis. And there's one producer, one major producer. There's others that are in process, and the bismuth-212 daughter presents some potential redistribution problems. Next slide. That concludes my presentation, and I appreciate your attendance and interest. Thank you.
Thank you, Dr. Atcher. At this time, we'll be conducting our question and answer session. Please hold for a brief moment while we pull for questions. So our first question comes from Faisal Khurshid from SVB Securities. Please go ahead.
Hey guys, thanks for putting on this great event. It was very informative. Just wanted to ask, so in terms of some of these up and coming therapeutic isotopes, which one of these kind of have enough supply today or sort of predictably within the near term to be able to supply enough doses for kind of mid to late stage studies or even commercialization?
Thanks, Faisal. Please go ahead, Howard.
Yeah, maybe I'll take this. I think, lutetium has a fairly robust supply at the moment. It would need to be, I think, ramped up to take on, a couple more approved therapeutic products in addition to Pluvicto and Lutathera. On the actinium-225 side, we have TerraPower that has the U.S. stockpile of thorium that should come online in the next year or so and be able to supply, I think, 2, maybe 3 products, and then that will top them out, and they have no capacity to expand given the nature of their production. There are several others coming online, but still to be proven. On the astatine side, as was mentioned, we have a relatively small production and a network of these alpha ion cyclotrons around the country, around the world. They're about 20-ish, but that's not scalable to wide scale use. as mentioned, we and others are looking at solutions for this. On the lead-212 side, there is a couple folks looking at this, and there seems to be a supply, and they're pushing forward on that now. So that's more to come.
Got it. And then just one follow-up on astatine and lead. I think you guys nicely laid out some of the theoretical benefits of those approaches. So for actinium, there were a number of FDA workshops and discussions and stuff with the working group to sort of discuss some of the considerations, such as around the 227 kind of co-production and implications of that. What types of discussions sort of need to happen with astatine and lead for it to kind of be ready for more of the prime time? And how far along are we in some of those discussions of the key considerations?
I think for astatine to be ready for prime time, the biggest issue is going to be wide scale availability of product. I think it's a well-known product. There are no co-products or long-lived half-life radioactive products to deal with. It decays to an inert product without any daughters. So I think that's a non-issue. And I think similarly with lead, just having a wider production. Astatine and lutetium both have potential for these co-products, actinium-225 and lutetium 177M, that are radiologic handling and potential radiation, excess radiation to the patients. That's why those discussions have to happen, and those issues don't exist in the astatine and lead.
Very helpful. Thanks for taking the questions and for putting on the event.
Thanks for the questions, Faisal. Our next question comes from Nicole Germino from Truist. Please go ahead, Nicole.
You're muted, Nicole.
Nicole, I see you're on the phone, so if you press *6, you should be able to unmute yourself.
Hello. Hi. Okay. Can you hear me now? Hey, guys.
Yep.
Thank you so much for hosting this again as well. And I got disconnected a little while ago, so forgive me if you had addressed this already. But given the short half-life of astatine, how do you envision using astatine-based radiotherapies as a viable commercial product? And how do you envision supplying it globally?
I think there are two potential solutions for that. Using the current technology of a relatively low current medium energy cyclotron. There could be a distribution network of these cyclotrons, much like the fluorine install base. That would require a fairly large installation effort, or domestically and globally, to make that happen. At Nusano, we feel we have a solution for that using our platform, given our very high production capabilities. And our plan is to have 3 or 4 facilities across the United States in order to be able to supply broadly the needs of astatine should several therapeutics come to market.
And just a quick follow-up. Can you give us any examples of tumor types where astatine-based radiotherapies would make sense?
Dr. Pomper?
Yep. Sure. I can't think of any tumor types, where it wouldn't make sense. Astatine has it's got about a 7-hour physical half-life. So you try to match the lifetime physical half-life of your isotope with the biological half-life of the agent that you're delivering. So I prefer working with small molecules because they tend to have fairly good pharmacokinetics. They tend to be excreted through the urine, et cetera, fairly quickly. So you really just need to time your astatine and your affinity agent to be around for about 21 hours, maybe a few days, maybe a day and a half. So it's very convenient, but we're using it now in prostate cancer and renal cell carcinoma. It's been used previously in glioblastoma, I believe. They're using it at Fred Hutchinson. They're using it at a couple places around the world right now. So I'm not sure where it wouldn't be. It's also chemically, because acetine is like iodine, all the same chemistry applies. So we can introduce it to aromatic rings, to small molecules. It's very easy to work with in that way. So I actually, as an organic chemist, I prefer that to having to, put on these big chelating agents where we then put the actinium or the lutetium or the radiometal. So you could tell by how I'm talking, I really like acetine, and it's, like I say, I think it's a kinder, gentler alpha emitter.
Great, thank you so much.
Thanks for the questions, Nicole. So our next question comes from Brandon Carney from B. Riley. Please go ahead, Brandon.
Hi, great presentation, very informative. Thanks for hosting, and thanks for taking our questions. I think it was mentioned some of the toxicity issues with the actinium-225 PSMA agents, specifically like the salivary glands, and just wondering, what efforts are being put into addressing those. Understand that maybe antibody targeting vectors might be helpful there, and I'm just wondering if isotope like acetine-211 can be used with larger targeting vectors like that.
Sure. So the efforts are, yes, using an antibody. Some are trying to use antibodies along with small molecules to try to leverage the advantage of both of these entities. Acetine has been connected to antibodies and used in the past. There's no issue with that. But I think that right now, what people are trying to do in Europe in particular is to try to change the dose rate or the dose scheduling, or how the patients are going to receive the agent, to try to cut back on some of these off-target effects. There are also efforts to develop agents that selectively block the salivary glands. But what we prefer doing is designing agents that just don't hit the salivary glands in the first place, as I showed on that slide. So there are agents that don't have that kind of off-target effect, or I guess they call it an on-target undesirable effect, because there is a little bit of PSMA in the salivary glands.
Great, thanks for that color. I guess maybe some more detail on the stability issues you mentioned with acetine-211. You mentioned that the chemistry is quite like the iodine-131, but I expect that the carbon-acetine bond is a little bit weaker. Just wondering how that compares and what kind of issues that might pose for acetine-based drugs.
Yeah, that's something that we always test for. In fact, generally we generate the compound with iodine, radioactive iodine first, and then we see how that looks. And then we try acetine. We do sometimes dual label experiments at the same time. And we will see, sometimes a little bit less uptake of the acetine based agent, because it's just frankly a bigger atom. Also, we can check very easily for if the acetine is somehow unstable and comes off the molecule by looking at a couple of organs like the stomach. It does the same thing iodine does, but we haven't really found too much trouble with the acetine in the PSMA compounds that we've made. We more or less need to have 95% or a greater stability or chemical purity and radiochemical purity before we inject into a patient. That'll be put a little bit to the test as we start sending this stuff around the country. Once Howard Lewin has his device working. But I think that the chemistry also is very well known, and it hasn't really proved to be a huge issue. In my experience, but again, I'm not an acetine chemist, but I haven't seen it really impact our preclinical studies just yet.
Great, thanks for that. And maybe one last question, maybe following on the supply and delivery question from earlier. Just wondering, with the short half-life of astatine, if it makes sense to maybe try to incorporate, even at these like centralized distribution sites like Nusano, maybe incorporate drug manufacturing into that, and if any thought has gone into that at Nusano.
No, absolutely. It's Chris, Brandon, and it absolutely, it is part of our plan. And I think you'll start hearing some very specific details about that. But the idea of having co-location with our facility 1 in Utah, I expect will absolutely be a part of the plan.
Yeah, right now we're doing a test run between Duke, which does have a high energy cyclotron to produce the acetine, and Johns Hopkins. So we're gonna probably either drive it or fly it up. And what they could do, they could potentially send us the target, which we can distill ourselves and do the chemistry ourselves if we have to get high specific activity material. So yeah, we're definitely pressure testing some of those issues.
Great. A lot more questions, but maybe I'll leave it there for now. Thanks again for hosting the event.
Thank you.
Thanks for the questions, Brandon. Our next question comes from Kemp Dolliver from Brookline. Please go ahead, Kemp.
Great, thanks for taking the questions and for hosting the event. First, it relates to actinium-225 that's accelerator produced and the levels of impurity of AC227. I mean, so far, DOE is indicating that they're able to produce it at levels of impurity that should be clinically acceptable. But what's your comfort level so far? Have you used any of this yourselves? Do you talk to your colleagues and have a sense of their willingness to use it?
Yeah, maybe Dr. Pomper, and then
I guess
Howard, do you want to comment on that?
I can comment a little bit. So we haven't used it clinically just yet. As Bob Atcher just alluded to, it takes a little getting comfortable with these alpha emitters in general. For our preclinical studies, I find that the astatine is it's very, shall we say, powerful. It's also very toxic. So we give portions of microcurie sometimes to the mice as our starting doses. But I think that if that can somehow, if they can get under control with respect to these impurities and the production, et cetera, then you've got the issues of the daughters, in vivo. So I'm not really sure. I'm not sure where acetine is or where actinium is going to be in our armamentarium, but I don't have a huge amount of experience with it, and I would have to defer to Bob or somebody else on the panel.
I can let me give you a little bit of what I typically suggest when we talk about the high energy production. They're irradiating a thorium target with the high energy protons, and it's not a direct production like you see with lower energy interactions. You're creating a fission event, which includes the production of actinium isotopes. And as a result, you've got the actinium-227 co-produced with the actinium-225. Realistically, my understanding is that those targets are so hot that they actually have to wait about 10 days in order to start processing them, which means that you've already lost one half-life of the actinium. And because you've got a number of other things being produced, which includes some lanthanide isotopes, the separation chemistry is much more complicated than milking a thorium-229 generator. And my understanding is that the chemistry that they developed takes about 10 days to get a good separation of the actinium for the other products that were made in that target. So you've got another half-life of actinium that you have lost. You will ship it out, and there's a high likelihood that people are not going to use it immediately on reception. So you're going to have yet another decay period, which can be probably as long as 10 days. So at this point, you've lost 3 half-lives, which means that the amount of actinium-225 that you've produced is down by a factor of 8. Whereas with a 20-year half-life, essentially there's no reduction in the amount of 227. Based on some of the published data on the co-production of the 227, I figure that you may have 2 to 3% of your material at the end of this process when you are now doing synthesis and preparing the radiopharmaceutical. You may have 2 to 3% of that material that is actually actinium-227 labeled. And the question becomes, when you look at the dosimetry, is that going to create enough issues relative to redistribution of the actinium in those patients that it's going to, the concern on the part of FDA may make it impossible to use those materials. So we still have yet to see appropriate studies, in my mind, that will make it, that would satisfy any concerns on the part of the FDA that you actually have that under control and that your 227 concentration is minimal enough.
Great. Thank you for those responses. For Nusano, it's mid-23. Your facility is going to open at the beginning of 2025. When do you start selling their production capacity, and how do you think about the offering? Because if you have 40 different options you can make available for people, that's a lot of potential decisions. Whereas today we're really only talking about, say, 2 to 4 isotopes. So can you give us some perspective from that angle?
Yeah, absolutely, Kemp. And that's the only thing that gives our chemistry group any stress is how quickly are we going to ramp up? We've actually started pre-selling production slots, and that's helping to dictate. I think a key part of our competitive advantage is the flexibility, as I mentioned. So our facility is going to produce up to 12 different isotopes at once. Now, obviously, we're not going to launch in Q1 of 25 with 12 right out of the gate. It'll be a gradual ramp up, but we've started that engagement with customers, and our goal is to be able to not only satisfy their commercial demand, but to be able to at least get minimum quantities of a diverse set of isotopes very quickly, so that we can support the ongoing or the soon to be ongoing clinical development programs.
Fabulous. Could you talk, since we've spent so much time talking about astatine, and I think that's where you've put some of your initial marketing effort, could you talk a little more detail about your plans for it to address the issues that have been discussed today?
Yeah, absolutely. And so in a couple of things, I mean, in terms of the short number one, first off, we'll have a co-location of complete manufacturing partners, which I think will be online in the 2025 timeframe. And again, more details on that will be forthcoming here very quickly, like literally in a matter of days or a couple of weeks. But in terms of the short half-life issue, I think, number one, our level of efficiency, our production capacity allows us to adjust for that in the manufacturing process, meaning we can allow for a certain amount of decay in the transit time. So while we might think of it as having historically a 7-8 hour half-life, we may be able to overcome that just with sheer volume of the product that we'll be able to ship. And I think doing that within the standard white glove logistics process that's, known in our industry. We think that that'll provide some good optionality for at least the customer base in the Western United States as it relates to facility one.
Great. Thank you for taking the questions.
Absolutely.
Thanks, Kemp. Our final question comes from Sylvan Turkin from JMP. Please go ahead, Sylvan.
Hi, thank you, and thanks for taking my questions and great presentations. I was just wondering, what are some of the key, I guess, challenges or let's say new scientific developments that we can look forward to with respect to conjugation chemistry, which would unlock significant applications for astatine, but also some of the other radioisotopes. Thank you.
Sure thing, Sylvan. Dr. Atcher, could I ask you to take a stab at that one?
Sure. As Dr. Pomper's pointed out, the conjugation chemistry for astatine is easy to test using iodine-labeled materials. And so I would say that as far as clinical application, the state of the art for the conjugation chemistry in that case is sufficient to eliminate that as a concern in terms of the application in theranostics. With respect to the metals, one of the things that I didn't talk about is that when an actinium isotope decays by an alpha emission, the recoil energy, and it's back to Newtonian physics in that there will be equal amount of energy that's distributed into the nucleus that's created. It by about a factor of 100 to 1,000. It is greater than a chemical bond. So anything that you have used to conjugate the actinium-225 to the targeting molecule is absolutely useless from that point onward, because your decay nucleus has gone off into a different direction, which is why there's concerns about the decay products in terms of non-target toxicity, because there's no guarantee that that isotope is going to stay at the site of the decay of the actinium-225. So you can make whatever you want. And I mean, the conjugation chemistry for the actinium is pretty mature. I know people will say that they're developing a better chelate for it, but it's kind of a moot point, because whatever whatever isotope you get downstream is no longer going to be conjugated to the targeting molecule. If it occurs within the cell, a very high likelihood that it's going to stay within the cell, and in some cases, even if it's if it's into a matrix of cells, maybe it will end up being captured by a neighboring cell. So that kind of covers it. I do want to back up for a second, though. We haven't talked about the fact that in the case of Nusano, they have the capability to accelerate not only the helium-4 nuclei, but also lithium-7. And you can then create a parent isotope of astatine-211, radon-211, which gives you the option of producing the radon and then shipping the radon to the site where the astatine will be utilized. And in transit, you've got the radon, which then is decaying into the astatine. So you essentially grow your astatine during shipment, and that provides an even more powerful potential utilization of astatine that is not necessarily limited by the distribution activity.
Thank you so much.
Thanks for the question, Sylvan. This concludes today's Q&A session. I'll now turn it back to Chris for closing remarks.
Yeah, first, I would just like to truly thank Dr. Atcher and Dr. Pomper for sharing their expertise and wisdom. There's no doubt that this is an evolving space and a space that we're going to be speaking about for many years to come. And I couldn't be prouder to be representing the Nusano team and delivering what we think is a breakthrough technology to help enable this next generation of radiotherapeutics. So thank you so much for joining today. And we look forward to engaging with you in the future. I guess one last thought is, I know there's a lot of online questions that we just didn't get a chance to get to in the abundance of time today. Please feel free to reach out to your LifeSci rep or directly to the company, directly to myself, and we will make sure that we get your questions answered and get anything you need. Appreciate your time. Thanks, everybody. Have a great day.